Hi everybody. Now let's talk about the discovery of the double helix of DNA. That's quite familiar to every one of us because in 1953, Watson and Crick put forward their double helical model. And we know that DNA is a double-stranded DNA and it has been wound on each other in the form of a helix. So let's go into the details of that. This is Watson and Crick. Watson and Crick, in fact, put the evidence together, which was being put forward by various other scientists, and then they proposed the Watson and Crick model, what we learned today. Chergav's realization that A pairs with T and cytosine pairs with guanine, combined with some crucially important X-ray crystallographic work which was being done by the English researchers Rosalind Franklin and Morris Wilkins, contributed to Watson and Crick's derivation of the 3D double helical model for the structure of DNA. Watson and Crick's discovery was made possible by the recent advances in model building and possibly the assembly of three-dimensional structures based upon known molecular distances and bond angles. A technique which was being put forward by an American biochemist, Linus Pauling. It can be seen that they were using cardboard cutouts representing the individual chemical components of the 4 bases and other nucleotide subunits, and Watson and Crick shifted their molecules around their desktops as though putting together a puzzle. So let's go into the details of it using an animation. So this is Watson and Crick, and in 1953, Francis and Watson and Crick, that's it, they published the first accurate model of the DNA molecule. They were interested in DNA, the hereditary molecule of life, and they wanted to build what was chemically known about DNA and to determine its actual structure. Now, the Phoebus Levin, he had put forward the structures, what are the constituents of DNA, that it was made up of adenine, guanine, thymine and cytosine, as well as the sugar. The deoxyribose sugar was a component, and we could also found phosphate groups attached to them to form a nucleotide molecule. So, keeping this in mind, they looked forward onto the structure of DNA, and different types of nucleotides could be attached to the phosphate sugar moiety, resulting in the formation of a nucleotide. So, and these nucleotides are linked in series from one phosphate to the next sugar to form the next phosphate and so on, and a polynucleotide was being formed. Although Levin had proposed the correct chemical linkages, his tetranucleotide theory was wrong. And if DNA had a fixed repetitive sequence, it wouldn't be intelligent enough to carry out information. And after Oswald Avery's landmark paper, they knew that DNA had to be intelligent. In fact, it made more sense if the order of the nucleotide could be changed and the information can then be coded in our DNA sequences. And DNA, but not protein, was the Rosetta Stone for unraveling the true secret of life. You can see the polynucleotides which were formed. Then Erwin Chergaff, he also contributed to their information, and he isolated DNA from different organisms and measured the levels of each of the nitrogenous bases, and here are his results. The amount of adenine is close to amount of thymine in all the samples which he analyzed. And when you looked, the amount of samples in guanine and cytosine is also being found to be the same. So he put forward the Chergav's rule, and that information was also imbibed by Watson and Crick. If Levin's tetranucleotide theory was correct, then the amounts of A, T, G and C would be the same in all the organisms. But this was not the case. So they concluded that the tetranucleotide theory put forward by Levin was not correct. The Chergav's rule, A pairs with T and G pairs with C. About the same time, Linus Pauling at Caltech used his knowledge of chemistry as a powerful technique called the X-ray crystallography to discover a corkscrew-shaped structure found in many proteins called the alpha helix. This was alpha helix. And Francis and Watson and Crick, that's it. They were following Pauling's approach of using chemistry and X-ray diffraction patterns to solve the structure of DNA. X-ray diffraction patterns can divide a lot of information about the shape and structure of the molecule. If a stream of X-ray is directed at a crystallized substance, some rays are diffracted or scattered as they encounter the atoms. The scattered X-rays then interfere with each other and produce spots at different intensities, and these can be recorded on a photographic film, as you can see here. Now, the resulting diffraction pattern is the unique signature of the molecule DNA. At the time, DNA couldn't be crystallized, but they could get two different types of DNA fibers, and these fibers gave two distinct diffraction patterns. You can see there were two different diffraction patterns for the two DNA fibers. So Rosalind Franklin and Morris Wilkins made these DNA X-ray diffraction patterns. She was the one who provided this diffraction patterns, and they concentrated now on the X-ray data from this form of DNA. As you can see, there are lots of more spots on it, and therefore more information was there. And using this, they tried to calculate the basic dimensions of the DNA molecule. While Rosalind was working on the other X-ray crystallograph, Francis was immediately struck by the symmetry of the simplicity of this X-ray pattern. It was clear that all the information they needed was in the X-ray pattern. The distance of the X in this X-ray photo is the telltale pattern of a helix. And because the X-ray pattern is so regular, the dimensions of the helix must be consistent. For example, the diameter of the helix stays the same. And in an X-ray diffraction pattern, the closer the spots, the larger the actual distance. So horizontal bars actually correspond to the helical turns, and the vertical distance bars 34 angstrom is the measure of the height of one helical turn. Now, they knew the height of one helical repeat was 34 angstroms, and they knew the distance between stacked base pairs to be as 3.4 angstroms, and there must be 10 nucleotides per helical repeat. So you should understand all this information was based upon the X-ray crystallographic studies which were being provided. Now the helix pitch, or the degree of rise, was also being calculated from the angle of X makes with the horizontal axis. Now if you distort the helix, you can get an idea how the helical pitch is related to the X-ray pattern. And from this X-ray diffraction pattern, they deduce the DNA should be a double helix with the phosphate groups on the outside and the bases on the inside. And from the measurements made by Francis and Wilkins, they knew that the basic dimensions of the helix. So they were again eager to fit everything known about DNA into an accurate model. But questions still remained: how do the helices fit together? How are the nitrogen bases arranged? So it was a race to solve the DNA structure, and then they knew that after solving the alpha helical structure in proteins, Linus Pauling was interested in the DNA structure. And just as they were beginning to build tentative DNA models, they heard that Pauling had submitted a paper on the structure of DNA. They waited on pins and needles to review his scheme, which turned out to be a triple helix. That was a triple helix, what Linus Pauling told. Everyone agreed that this model couldn't be right, that is, Pauling's triple model was not possible, and Pauling put the phosphate groups in the core of each helix with nitrogenous bases facing out, and 3 such helices that then intertwined to make the DNA molecule, but that couldn't be correct. And Pauling had forgotten the negative charges of the oxygen atom in each phosphate group. Facing towards the middle and stacked up on top of each other, these charges would repel one another, making it impossible for the molecule to hold together. And almost unbelievably, the man who had written the book on chemical bond got it wrong, and Linus' mistakes encouraged them to work even harder, and they knew he would redouble his efforts to set the error right. One day, not long after his paper came out, they began to play with paper cutouts of nitrogen bases. These were the nitrogen bases, and they knew that nucleotides could pair and form weak bonds called the hydrogen bonds. Unlike the strong covalent bonds that join the phosphate sugar and base into the nucleotide, hydrogen bonds are formed when hydrogen, nitrogen or oxygen shares a hydrogen atom. And they started pairing it, and they looked what all possible combinations are there. And it was being looked. When they tried, they found that the possible thing was probably, according to the different hydrogen bond pairs, some pairs were very different in their width. And if these pairs really occurred in DNA, the helix would be uneven and wouldn't bulk in and out. So they realized that adenine could pair closely with thymine and guanine could pair closely with cytosine, and the AT base pair was the same width as its GC base pair. So that was the only possible solution by which the DNA could be closely packed within it. And this base pairing agreed with Cherkav's ratio and allowed the bases to compactly stack on one over the other. They were convinced that the base pairing was a key to DNA structure, and they pointed out that because of the certain bond angles and the proximity of the base pairs, the two helices had to run in opposite direction, and then that means that the helices were anti-parallel to one. So this is the illustration adapted from the double helix. And using metal scraps from the machine shop, Francis and Watson and Crick, they built a 3-dimensional model of DNA, and this 6-foot model incorporated what was already known with the AT and the GC pairing scheme and the Francis idea of anti-parallel strands. This is the DNA model, which is shown there in the pic. And everything clicked into place beautifully, and now we do learn that everyone, including Morris and Rosalind, agreed that they had the right DNA structure. So the Watson and Crick model was being proposed with the ideas of various others. The experiments which were being conducted by various scientists helped them to bridge on to the Watson and Crick model. So with this, we come to an end of the session. Thank you.